Gregg Semenza, born in 1956 in New York City, is a pioneering scientist whose work has profoundly impacted our understanding of cellular response to oxygen availability. Semenza began. Gregg Semenza, born in 1956 in New York City, is a pioneering scientist whose work has profoundly impacted our understanding of cellular response to oxygen availability. Semenza began. Gregg Semenza,
We continue with application of Professor Gregg L. Semenza research. Professor Gregg L. Semenza research on hypoxia inducible factor (HIF) has profound implications across various fields of medicine and biology. Below are some of the significant applications and benefits derived from his work. Application 1 is cancer therapy, targeted tumor hypoxia. What is this? This is HIF-1 inhibitor. Semenza's work has led to the development of drugs targeting HIF-1, aiming to inhibit its activity in hypoxic tumors. This can prevent cancer cells from adapting to low oxygen levels, reducing their survival and growth. Combination therapies. HIF-1 inhibitor can be combined with existing treatments like chemotherapy and radiotherapy, potentially enhancing their efficacy by making cancer cells more susceptible to this treatment. Under cancer therapy, we also have understanding cancer metabolism. Meta-reprogramming is by elucidating how HIF-1 regulates genes involved in metabolism. New strategies can be devised to disrupt the altered metabolic pathway in cancer cells, hindering their energy production and growth. The second application is research and diagnostic tools. Biomarker development. Biomarker development is hypoxia marker. HIF-1 regulate gene and proteins can serve as biomarker for hypoxia in various diseases, aiding in diagnosis and monitoring of disease progresses and treatment response. Next, we have research advancement is cellular and molecular biology. For this, Semenza discoveries provide fundamental insight into cellular response to hypoxia, driving further research and innovation in cell biology and related fields.
Gregg L. Semenza invented many advantages such as scientific understanding. He discusses how tackling fundamental questions in biology can lead to important discoveries that impact our understanding of human disease. He also improved public health and policy. He reminds us of education and awareness by increasing the understanding of hypoxia-related diseases can lead to better public awareness and preventive measures.
Medical advancement is the next benefit of Gregg L. Semenza's study. The hypoxia inducible factor or HIF pathway was discovered by Semenza, and this finding completely changed how anemia is treated. Based on Semenza's research, recombinant EPO therapy is frequently used to treat anemia brought on by chemotherapy for cancer, chronic renal disease, and other illnesses. Next, one of Gregg Semenza's research advantages is biotechnological innovation. Based on Semenza's study, recombinant erythropoietin or EPO therapy is a well-known example of a treatment for anemia linked to a number of illnesses. Biotechnology firms are also looking into other HIF-related proteins and processes as possible targets for developing drugs for conditions like cancer, heart disease, and other conditions.
The advantage of Gregg Semenza linking HIF-1 to cancer. To extend the research, Semenza worked again with Gregg to create a mouse embryo system cell that lack HIF-1 and therefore cannot produce a function transcription factor. Semenza found that the embryo not normally and stable grow up and the low oxygen. The knockout embryo failed to continue development and display major malfunction, especially in the candidial system. In HIF-1, the failure of the normal development of the heart, the blood reserves, and the blood are the three components of the pulmonary system fail and the embryo die. Number 2, the common cancer component. The outcome led Semenza and other scientists to conclude that HIF-1 has multiple roles in the formation of tumor, and it does in development. It can be included by genetic alteration and low oxygen condition. The combination of the speedway lead to very high HIF level, which we now believe to be a final common component which many of the genetic can be affect cancer biology. Number 3, Nobel Prize in Personal Medicine in 2019 withdraw because the author of the concept manuscript have some degree responsibility. Semenza be the senior author, so many of the paper has had responsibility to ensure the work is solid and legitimate. Semenza has repeatedly failed in this important task, and Semenza did reply to rescue of for the comment because many paper not income serious problem in his lab.
Here is how case study and articles will help to improve genetic, molecular biology, and medicine. First, gene regulation and cellular adaptation to hypoxia. Second, it will increase knowledge of cell auto-sensing capabilities. Third, overrepresented molecular pathways related to oxygen homeostasis, angiogenesis, metabolism, and cell survival. Next, open the doors to new therapies aiming at inhibiting HIF-1 signaling for multiple diseases. Next, known abnormal HIF-1 signaling is one of the hallmarks for development as well as progression and metastasis in cancer. This inspired the further development of HIF-1 inhibitors as potential anti-cancer agent. Moreover, enriched concept for tissue repair strategies in cardiovascular and respiratory diseases. Last but not least, provides a unique perspective on research advance in molecular pathogenesis and therapeutics.
For the case study, pharmacology targeting of hypoxia induced cyber factors. The ability to develop and maintain the structure and function of complex metazoan organism is dependent upon the ability of cells to efficiently generate ATP by oxidative phosphorylation. All metazoan species other than Porifera, means sponge, and Ctenophora, comb jellies, utilize hypoxia inducible factors (HIF) to maintain oxygen homeostasis.
In essence, Dr. Gregg Semenza's many awards and honors include the Nobel Prize in Psychology or Medicine in 2019 for the discovery of the mechanism whereby cells detect and adapt to levels of oxygen, a finding essential for progress in our understanding of cellular biology. His innovativeness in methods and revolutionary findings inspire further research in the struggle to understand the intricacies of human biology and to find new ways of managing a great number of diseases.